Thank you everyone for coming here, and I would like to thank organizers for inviting me. I heard about the conference of Uyghur Academy last year, and so I'm really pleased that I was able to come. Unfortunately, I can only give a presentation in English, specifically when it comes to science. You know, I don't know how to say many terms in Uyghur. But I'll try to kind of be very slow, and I know most of you speak English, so should be understandable. So some of my slides are maybe a little bit technical because I just came from a conference where I presented some of this data, but I'll try to explain in more simple terms. I have been working in kind of two major areas of research. One is in stem cells, and particularly we try to make new stem cells that usually our body doesn't have. The other, my research area is to study the gene defects that cause sometimes aging, and that specifically concerns to mitochondrial genes. So the mitochondria are organelles in each cell, and they have actually their own genome that's usually inside of this mitochondria. It's called mitochondrial DNA. And so we as the humans, and actually most of the animals, will have two different genomes. One is nuclear genome, which contains most of the genes that we know about. But the mitochondrial genes are very separate because they reside in cell cytoplasm, and it's very small and circular DNA. But their function is very important because they provide energy for everything we do. So basically, while I'll be speaking to you, I'll be breathing the air, and I have to do it like very often because my mitochondria need this oxygen. So what mitochondria does, it takes this oxygen and of course the food that we eat in form of, for example, glucose, and it converts into the molecule, it's called ATP, which it's kind of universal molecule that now our body and cells can use to basically to every function. So the mitochondria provide very basic function that basically support the life. Mitochondrial DNA is, as I said, very different, and we actually just learning, how this mitochondrial DNA functions. Actually it's been discovered pretty, late compared to nuclear genome. We inherit our mother's mitochondrial genome because it's only oocyte or egg provides the mitochondrial genome, where for some reason sperm doesn't contribute any. Another interesting thing is that the mitochondrial genome, since we don't have paternal genes, so there is no recombination, there is no heterozygosity, and it's considered that mitochondrial genome is passed from one generation to another without any change, even though it's not exactly true. And at least in my lab, we study the changes that occur in mitochondrial genome in eggs, and how this mitochondrial genome evolves with evolution in species. Interesting thing is the mitochondrial. Each cell has 1,000 mitochondria, and that's how we could have up to 10,000 copies of mitochondrial genome in each cell. So the mitochondrial DNA has been known to prone to mutations, so they mutate much more often than nuclear genes, and that's why there are a variety of tissues will accumulate mutations in mitochondrial DNA, and it's considered that actually its processes depends on the level of energy used. So many tissues that use lots of energy, for example, it could be heart and brain, they actually get more damage to the mitochondria, and they age faster, and that's how the primary causes of the aging or age-related disease are in those tissues. It could be heart attack or stroke. And it's considered that the mitochondria are in basically that's what's causing these conditions. We also study some of the genes, mitochondrial genes, that mutate in eggs, and that's how they pass from mother to children. So the mitochondrial gene mutations actually affect every part of the body, just because they constitutively expressed everywhere, because every cell needs energy. But they primarily strike the tissues that use most of the energy, and it happens to be brain, it's at heart, and the other vital tissues and organs. So when mutations occur in eggs, basically it's inherited gene mutations. It's really difficult to predict, how they will be passed, just because mutations occur not like in every copy of mitochondrial DNA, usually in the part, and it's called the heteroplasm. It's basically a mixture of two different mitochondrial types, mutated and healthy. And that ratio always changes, and that's how, for example, this mother, she was affected, in red, but she passed it to her two daughters that had very low mutation load. Well, basically just 20%, 25% mutation of 44, and these daughters were phenotypically healthy, but they passed to next generation more mutated copies, and for example, this son had 85%. And most of the time when the threshold reaches more than 50%, sometimes more than 60%, that's where the disease usually appears. So the inheritance is very complex, and it's impossible to predict whether the next child will be healthy or not. So we've been trying to come up with a solution. For example, if a family has this gene mutation, and for example, they already had one child that's been affected, can they have another child healthy? And the way we thought is, we don't have to kind of basically try to predict, but we have to go and treat these mutations before actually it's passed to the embryo. And the way we did it is that since this mitochondrial genome is passed from the egg, so basically a female will provide the egg, and sperm, as I said, does not contribute any mitochondria. All these mitochondria will be contributed to the offspring. So we come up with the idea that we could replace the mitochondrial genome from one egg. For example, this is mutated egg, then it has all this mitochondria mutated. But we could remove the nucleus and throw away the cytoplasm. That's where the mutated genes are. But the donor mitochondria would have to come from another egg. So that was the whole idea, that we don't have to make these artificial genes, but we could just take it from another egg donor, just like a tissue donation or organ donation. So it's very similar, but in this case it's gene donation. So this was the concept we've been working for many years, actually, and of course initially we tested in animals, and so we showed that this procedure can be done, and so now you can fertilize this egg with sperm, and then transplant this embryo into monkey, and these are our first monkeys that were born in 2009, Mita and Tracker. So they've been both born after basically completely replacing their mutated DNA. So since then we produced several more, and this is when they were born, and this is when they're 3 years old, and we showed that now they don't have any disease, they're healthy, and of course the mitochondria genome is completely replaced, it's healthy, and there is no recurrence of these mutations. So this was kind of work that we've done in rhesus macaque, and we published it in 2009. And of course we proposed it. Now this treatment probably will work for human because of, we work with monkeys, and monkeys are considered non-human primates and very close species to us, and usually whatever works in monkeys you can easily translate to humans. Even though it's a very expensive model to work. But I've been blessed to work at one of the largest non-human primate research centers in Oregon, where we have, of course, access to thousands monkeys. So once we did this, we decided that we need to replicate at least a portion of this work with human eggs. In this case, we didn't have completely approval to transplant this embryo into patient, but we could create these embryos in vitro, and basically it was the same. We would take it from patient carrying mutations, and then healthy egg donor would provide the cytoplasm or healthy mitochondria. And we did this replacement and also showed that now this embryo looks healthy with completely new mitochondria genome, and we made actually stem cells instead of babies. And we did all these tests on stem cells. So the title of this paper that we published this year in Nature was Toward Germline Gene Therapy. So it's a gene therapy of inherited diseases, and it's because we're dealing with germ cells, the cells that actually are passed to the next generation. It has some, the ethical concerns because procedures like that actually banned in many countries, just because I think when scientists actually developed this recombinant DNA transgenic animals, so the society and people started worrying that some days it will come to humans and we will be genetically engineering humans, enhancing. Even though this procedure is developed for basically treating mutations that cause terrible diseases, but there are still rules that we have to go through. For example, as I said, in many countries the law prohibits any genetic manipulation of the germline. That, for example, in UK. But since the procedure has been developed and we show the efficacy, so now governments are looking into changing the law. So in the United Kingdom, they've been looking for second year now to change the law just because we have a procedure. And they're changing specifically to these mitochondrial gene mutations just because this seems like it's going to work. And it's considered that probably sometimes next year the UK will be first country approving this kind of procedure. So I'm very proud that we took it from basically from the concept, and we did it in animal model, and then we moved to human. Basically, so we consider this is phase 1 study on safety and efficacy. So in United States, I also approached the FDA, and we proposed that we start clinical trials as well. And the end of this month, we're going to have a committee hearing where they're going to basically decide how they're going to regulate these clinical trials and what the condition is going to be. But I'm very hopeful that we'll start the clinical trials sometimes next year. And so idea is that now these embryos will be transplanted into patients, and we will monitor health of the children and up to maybe 3 to 5 years after birth. So this would be first in history when we basically cure genetic diseases, and basically before they happen. So my other part of the lab, actually working with stem cells, is, particularly we work with kind of early embryonic cells. It's one of the early stem cells that can make every other cell type. Initially, these cells were established as a cell line from early embryo, and it was done in '98 by my colleague, also primatologist, who used to work at the same primate center where I am now. So he's done it first in rhesus macaque, of course, as a model, and then he did it in humans. So these cells can be cultured to like early stem cells, and they don't differentiate unless you allow them. But you could multiply them to many passages and make lots of cells. But then you could induce them to differentiate into all these cell types, and of course many of these cells are needed for these patients. For example, especially if it's age-related disease. And if it's a Parkinson or diabetes, now we have cells that we could transplant to the patient. But the problem was that the immune compatibility, as you know, this came from unrelated embryo, and it wouldn't match to the patient that needs these cells. Of course, we don't have these stem cells in our body. We wear these stem cells, right after fertilization, but these stem cells during normal development, they differentiate into our body parts. So that was kind of one of the main obstacles, how to use these cells for treating patients. There was actually in 2006 when Japanese scientist Shinya Yamanaka from Kyoto University, he actually found a way to make artificial stem cells. In this case, he would just take any cell type from the body, usually sometimes it's skin cells, and then by transducing them with several genes, and these are kind of set of genes that he used, he can actually turn them into cells that just look like embryo-like stem cells. So he called them pluripotent cells, induced pluripotent cells, or IPS. So it's a very promising field, and it's been, basically used since then in thousand labs. And he was awarded Nobel Prize for that last year. So he's a prominent Japanese scientist. So now it seems like we have stem cells that will match to this patient. But unfortunately, for the last few years, when we study these cells, it seems like they have lots of genetic abnormalities compared to normal embryonic stem cells. So they have mutations that presumably caused during this kind of conversion. And also there are some, the epigenetic profiles. They have a memory that they came from these skin cells that don't exactly match to embryo-derived stem cells. So those issues actually now not allowing to move these cells, or propose these cells for treatment of patients. So the IPS cells been mainly used just the Petri dish kind of disease model. So it seems like we still need clinical grade histocompatible stem cells. And so in my lab, we actually been trying to derive a new class of stem cells. This time we use kind of different approach. It's called somatic cell nuclear transfer. And the way we do it, we will take, for example, any somatic cell from a patient, just like Yamanaka did, and this could be a skin cell. But we don't do this kind of genetic induction of pluripotency, but what we do is we take a nucleus, but we put it into egg cytoplasm. So it's unfertilized egg. And for some reason, the human egg cytoplasm has factors that we still don't know which one are they. They can induce change in this skin cell and make it like an early embryo, and then you could make embryonic stem cells. So these factors seems like universal in every species. That's how the cloning of Dolly has been done. But of course, in this case, we were not intending to clone humans, but to make stem cells, and even though the people still call them cloned stem cells. So this type of work has been envisioned almost 15 years ago when Jamie Thompson first developed human stem cells. That's when everybody thought this is a way you could make stem cells, but it didn't work for so many years. But in my lab, we've been working on this for the last 10, 15 years. So this is kind of my expertise area, to understand what's in cytoplasm that induces reprogramming, how support this reprogramming. And of course, we also used initially monkeys. We made similar stem cells in monkeys in 2007, the same year when Yamanaka made his induced pluripotent cells. But that time I was in a monkey, and it took us like 5 years to translate it to human. So we had to adapt. And several months ago, we published in Cell the first kind of proof of concept. Now we could make these stem cells. Of course, you need all the eggs to make these cells. So that's kind of negative. So we always need eggs. But we made it so efficiently that basically you only need like 2 eggs to make 1 cell line, which is incredibly high efficiency. And it doesn't seem like it matters. We could make it from very young patient, or we could make it from a patient that like 72 years old and has a disease, a neurological disease. But most importantly, of course, now we made the stem cells. Everybody was asking, are these cells the ones that everybody was looking? Are these clinical grade? And so the last few months, we've been comparing them to these Yamanaka cells, our cells. So basically, they came from the same source, from the same skin, from the same patient. But we would make it using Yamanaka technique, or we would make it our own. And we've been comparing, especially those genetic mutations that we know the iPS cells have. So we use the platform, it's called SNP genotyping, that actually looks for almost 5 million different loci in human genome and compares them. And basically what it shows is actually we see that those Yamanaka cells have a very high number of these abnormalities. So basically it's more than 2 per each cell line, all kind of abnormalities. So our clone stem cells have much less, so basically 3-fold less. And it shows that these cells, at least, we made 4 cell lines. Only 2 of them had kind of minor abnormalities, but 2 were very clean. And this is the first time we could derive such a clean genetically cells that have no kind of mutations or arrangements. Another thing is, as I said, the epigenetically, the level of reprogramming. So the induced pluripotent cells, they always have a memory that, for example, if they came from skin cell, they kind of resemble skin cell. And these are kind of, the way we do it, we do global methylation profiling. That's one of the ways to look at the epigenetics. And of course, we've been comparing our clone cells to iPS cells, and you could see in various regions of the chromosomes. This one is on X chromosomes, so the level of reprogramming very complete. So our cells look like real embryo-derived stem cells. We've looked at a variety of different regions where you could always see that nuclear transfer cells stay very close to embryo-derived, where the iPS always have the kind of abnormally reprogrammed regions. We also looked at the gene expression using this kind of new way of what's called RNA-seq, and we also showed that these clone cells are very similar to embryo-derived. So this is kind of, I think, 4 cell lines, and this is 2 controls. These are iPS cells, this is Yamanaka cells, and they're quite different in a variety of regions. So basically, we haven't still published this paper, but everybody is expect to show these cells, are they really clinical grade? Can they be now used for transplantation? And so far, they looks like, yes, it's a real cells, they just like embryo-derived, even though the nucleus came from patient, and that means they're histocompatible. So we've been able to take best from 2 different cell types that existed before. We had real embryonic cells, but with patient's own nucleus. And looks like, we got these cells now, and hopefully this can now move to clinics faster. So basically we prepared now this paper, and we're showing, all these differences between these two different cell types. So the conclusions are, basically epigenetically and transcriptionally, our clone cells are very close to genuine, embryo-derived stem cells from in vitro fertilization. And we also see that iPS cells have very high genomic abnormalities, and our clone stem cells have very little. And so we hope that these cells will be much easier now, for example, to approve through FDA as a therapeutic agent. So that's kind of the second part of my lab, and we've been working, as I said, these cloning techniques for years. And the main idea was, of course, now we could produce high-quality stem cells that now probably we could use it for therapeutic purposes. So this is basically, we use eggs, unfortunately, but you get high-quality cell, which is a clinical grade. So I have a bunch of people that work in my lab. This is a members of my lab, but I also collaborate with lots of clinical departments. So, for example, when we need eggs, so we have a department of OBGYN. That's where they enroll patients and they retrieve eggs. So I enjoy funding from NIH. But unfortunately, NIH only funds animal studies. But unfortunately, NIH does not fund human studies, and so we have several non-federal funding. Some of them are private funding that support my lab. So that's basically all in short. Hopefully it wasn't too technical, but if you have any questions, I would be happy to entertain. Yes, please.
Bilimdek bir